Welcome to our group's presentation on GFP. In 2008, Shimimura, Chalfie and Chen won the 2008 Nobel Prize for their work for the discovery and development of GFP. Shimimura could not have predicted the far-reaching discoveries that would take place in the following years after he first isolated GFP from the jellyfish Aquaria victoria and discovered that this protein glows bright green under ultraviolet light. The purpose of GFP is to highlight proteins in live cells. GFP has many uses, which include fusion tagging, which is one of the most common uses. GFP can be fused to the N- or C- terminus of a protein, which allows the scientists to visualize when and where the gene is expressed. Transcription reporter. Placing GFP under the control of a promoter of interest can be used to effectively monitor gene expression from that promoter in a given cell type. This type of transaction reporting was amongst the earliest uses of GFP. FRET, or Foster resonance energy transfer. This is used to study the interactions between two proteins or between two domains of a protein that undergoes conformational change. Typically, two fluorescent proteins with overlapping excitation emission spectra are used. One fused protein or domain being tested. Cell marking selection. Expression constructs like plasmids often include GFP as a marker to help identify which cells have successfully taken up their plasmid. FACS, or fluorescence activated cell sorting. This is a type of flow symmetry that separates mixtures of cells into distinct populations based on fluorescent signal. FACS can be used to separate cells expressing GFP from cells that are not.
Please dim the light, please. I apply UV light.
The history and development of GFP covers a period from 1955 up until 2008 when Chalfie, Shimimura and Chen were presented with the Nobel Prize for Chemistry. Of course, research is still taking place, finding more uses for GFP. When presented with the award, the discovered glowing luminescent protein was described as a guiding star for biochemistry. The main scientific contribution of Shimimura was the study and understanding of the bioluminescence mechanism of Aquaria victoria jellyfish. When Shimimura was presented with the Nobel Prize in 2008 in Stockholm, he demonstrated luminescence of GFP with an ultraviolet blue light. You can see this in the video to the top right. Martin Chalfie, who learned about the existence of GFP for the first time in 1988, realized that GFP would be a fantastic tool for mapping cells by connecting its gene with those encoding for other proteins. In 1992, Douglas Prasher cloned and identified the nucleotide sequence of wild-type GFP, and in 1994, Martin Chalfie expressed its coding sequence in E. coli and C. elegans. You can see this in the video to the bottom right. In 1995, Roger Chen engineered the wild-type GFP, obtaining mutants with different emission wavelengths and improved thermostability. Later, enhanced GFP was prepared. Other blue mutants, cyan fluorescent proteins, and yellow fluorescent protein derivatives were also constructed and characterized. Chen could not produce red with GFP. This proved to be very important as red light penetrates biological tissues more easily and is very useful to researchers who study body cells and organs. Mikhail Matz and Sergei Lukyanov searched for GFP-like proteins in fluorescent corals and discovered 6 more proteins, one red, one blue, and the others green. Chen's research group redesigned DS-Red so that the protein is now stable and fluoresces as a single amino acid chain that can easily be connected to other proteins by genetic fusion. Chen developed other proteins and named them according to the color and glow. You can see a beach scene here drawn with fluorescent proteins derived from GFP and red fluorescent coral proteins. Here we can see the 3 2008 Nobel laureates in chemistry. After being presented with the Nobel Prize for the 50 years of research work, starting with the bioluminescence as a natural phenomenon in living marine organisms to the development of powerful tools to study and quantify molecular events in complex biological systems. Unfortunately, Roger Chen passed away on August 24, 2016, after a lifetime of groundbreaking research.
Hi, I'm Isabel, and I'll round off this discussion on green fluorescent proteins. I'll discuss the advantages, shortcomings of the technique, and finish with a quick look at the future trends in fluorescent proteins. Green fluorescent proteins are tiny. They can be added to a particular gene or tagged to a specific structure or organelle within a cell and not interfere with its activity. It simply makes whatever it has an affinity for glow when activated. It is programmable using antibodies. Ready-made GFP variants are available on the market that have affinities to specific proteins or organelles. Alternatively, it's inserted into a gene as explained by John, included before the stop codon in a sequence. When that particular gene is activated or expressed, it glows, making it a reporter gene. GFP and its variants are non-phototoxic. They do not kill or interfere with the protein they are attached to, allowing scientists to observe how live cells work, how their organelles carry out functions, how DNA unfolds and replicates, and how this information is inherited, and also the consequences of a particular gene or protein being impeded or turned off. Scientists are able to implant GFP into live animals to track diseases such as Alzheimer's, HIV, malaria, heart disease, to better understand how these diseases move and affect different organs and systems within an organism. Photostability refers to the length of time the luminescence can be observed. It's sometimes referred to as photobleaching, how long before the glow bleaches or fades. This is caused by the degradation of the protein every time it is excited. As it returns the energy, the glow, part of the particle is worn away. Photostability is important when taking images of processes in live cells where multiple images are required over a longer period to capture the full process. Photobleaching is relatively low compared to other products on the market like Fluorescent. Anti-fade reagents can be used to lengthen photostability. The easily recognizable fluorescent glow with a predictable excitation extinction range helps to quickly and reliably pinpoint the target under a microscope. Having good compatibility with other dyes or fluorophores, as well as the same product available in different colors, allows for multiple tagging in the same cell. For example, multiple organelles or proteins can be tagged and imaged at the same time to provide greater insights into live cell activity and interactions. No other chemicals are required to make GFP glow. Alternatives like aquarium and luciferase require additives to activate them. GFP is easy to use and is naturally available, requiring no additional processing or purification before use. GFP can be used across a number of imaging and microscopy techniques such as fluorescent microscopes, confocal microscopes, and flow cytometry, and in macrophotography where a whole organism can be imaged. Shortcomings. Cytotoxicity is a problem. The product can kill if the cells are overexposed or reduce their ability to regenerate. This is lower than other techniques, but still possible. GFP tends to glow more brightly at lower temperatures, so the glow is less in mammalian live samples than in plants or fixed samples. It also photobleaches, fading after a period of time when taking more than 10 images. The bright light used for the excitation of fluorescence can generate free radicals, which may damage cell proteins or break the DNA strand. The brightness of some GFP mutants may be sensitive to changes in pH. The barrel-shaped structure, as shown in the image, is essential to the fluorescence. When developing mutants, new colors of GFP, it was discovered that any change to the structure, particularly the protective beta helix shape, stopped the glow. There are many alternative luminescent products available now, some based on GFP, and many use GFP as the standard comparative for their products. Crystal ball gazing isn't my forte, but here goes. Scientists have already made much headway in understanding microbiology, disease, metabolic processes, and genes. Physics, physiology, chemistry, genetics, anatomy, and biochemistry were areas that Dr. Chen identified back in 2003 would greatly benefit from the technology. They have and will continue to do so in the future. It has also sparked some new industries such as nanobiology, nanomedicine, and nanobiotechnology. As a group, John, Suba, and I believe that GFP will help in breakthroughs in understanding and overcoming known and new diseases and in gene therapy by eliminating hereditary diseases, advances in neurology, and industrial uses, such as the already existing identification of landmines in mine clearing. Bacteria able to identify the chemicals used in explosives are tagged with GFP, then sprayed on an uncleared minefield. Readings are taken with a microscope, which looks much like a telescope, allowing mines to be identified up to 50 metres away, much safer than many current techniques. Pharmaceutical companies benefit from medical breakthroughs with better understanding of the complex interactions that occur within our cells and bodies, and identifying active ingredients in plants and animals, for example, lipids in bilirubin affinity in eels. These may lead to advancements in drugs and food development, agriculture, and hopefully healthier environments. Cryobiology, such as cryogenetics, freeze-drying, and cryosurgery will improve. Forensic science will become more precise, and gene technology will also be at the forefront. Many animals, or more animals, will be mutated to glow, such as the already available glowfish, to create a new trend in pets. More fluorophores will be discovered, such as the latest Q dots, quantum dots, which are fluorescent but even smaller than GFP and used for nanobiology. However, these currently impede the function of proteins. Mineral-based fluorophores are also available with shorter protocols, reliable tagging, faster results, and greater photostability. As in all industries, economic return will drive the discovery of many new fluorophores for highly specific purposes. The Molecular Probes Handbook already lists over 3,000 molecular probes and kits, and there's no telling how many more will be discovered as we master the fluorescent. As discussed throughout this presentation, GFP has transformed how we understand the world. With it, scientists have revolutionized cell biology. We can now show how diseases work and identify ways to stop their progress. We can pinpoint genetic diseases and are at the cusp of turning off the genes responsible. Our understanding of metabolism and metabolic diseases is so much greater thanks to the inroads created by GFP. The works of Dr. Shimura, Chaufey, and Chen and their teams on GFP were recognized by the award of the Nobel Peace Prize in 2008, by which time many uses for GFP had been identified. Understanding how GFP works, as discussed by Suba, has allowed it to be mutated into a whole spectrum of colours. This extends the usefulness by allowing multiple tags to be set and imaged or contrasted against other colours. The GFP revolution is not restricted to cell biology. It's provided a means to scientifically test biological theories, extend this knowledge to understand full organisms. It's provided useful insights in many industries, in pharmaceutical, medicine, biology, chemistry, neurology, the nano world, and is the grandparent of a whole host of new and exciting fluorescent dyes that will continue to improve our understanding in the future. Thank you.